as neurons are. And also Santiago Ramon y Cajal already made very nice drawings about glial cells in the brain. And here you see the nice astrocytes interacting with neurons, and also cells he called the third element, which are also interacting with neurons, which are the microglia cells and also the oligodendrocytes. In the brain are several types of glial cells. You can call them the microglia and the macroglia. So the microglia are mainly the immune cells of the brain. They're also from a different lineage, developmental lineage. They are from the hematopoietic stem cells, and then it becomes important if you think about making cerebral organoids. And then you have the macroglia. Here you see the oligodendrocytes, of course, important for forming the myelin. And you have the astrocytes, which have a multitude of functions, are directly interacting with synapses, but also directly interacting with the vasculature in the brain. So in many neurological diseases, there is a very clear change in glia, in astrocytes and in microglia. So normally these cells are looking like what you see here in the picture, with a lot of processes here in microglia. But at the moment when there is a disease process ongoing, the astrocytes become reactive. So that means they become hypertrophic. There's a huge upregulation of cytoskeletal elements such as the GFAP protein. And you see that also the microglia become activated, meaning that they round up, they get a more macrophage-like phenotype. And what I'm really interested in is how that also affects synaptic function, because both cell types can also interact with synapses, and how that change in the glia, which is a response on the disease process, could affect synaptic dysfunction and cognition. So if we zoom into one synapse, the presynaptic and a postsynaptic component, we see that astrocytes are really enwrapping the synapse, are closely interacting with the synapse, and the microglia are constantly moving around and looking in the environment to see whether there is something that they need to react to, like viruses or bacteria, but also amyloid, for instance. And what we know also from studies of Beth Stevens is that, for instance, in Alzheimer's disease, in an Alzheimer mouse model, those microglia can start eating away synapses, which is called pruning. And that's what you see here in these two pictures. So the green and the red are the puncta for the post and presynaptic elements. And in an Alzheimer mouse here on the right, the G20 mouse, you see that there are a lower number of these puncta. And that's also what they quantified, indicating, and they also show that in the paper, that synapses got lost and that microglia are involved in that, and they are actively attacking the synapses. And in that way, they could affect also cognition and neural circuits, for instance. On the other hand, the astrocytes, and there's a lot of work done on that, they form the so-called tripartite synapse. And in this work of Henneberger, already 10 years ago now, what they have shown is that if you produce an LTP, so you give a high frequency stimulation and you measure the potency of a synapse, after a high frequency stimulation, the synapse is more potent, so you get a higher signal. But at the moment, if you prevent an astrocyte to react to this synaptic transmission by calcium chelating, what you see, there's no LTP. So this shows that astrocytes can really affect LTP, can affect neuronal interaction, and also can have an effect, for instance, on learning and memory. And if you think about that, one astrocyte in the human brain, it's calculated that one astrocyte is interacting with about 2 million synapses. And if this astrocyte is changing in a disease process, it can affect many, many different synapses, and therefore should also be taken into account in how that could affect neural communication and circuits. So I'll show you a little bit of our Alzheimer work, which we have already published several years ago. And then I would like to make the move to the more human models. So in Alzheimer's disease, which is the main form of dementia, there are a few very clear hallmarks if you look into the brain. So the human brain is much smaller. It lost a lot of weight. You also see that these gyri are becoming much wider. And if you look under the microscope, you see amyloid plaques, which are extracellular deposits of amyloid beta peptide. you see that in neurons another protein accumulates, which is hyperphosphorylated tau, and then this forms the tangles, but there's also a clear reaction of the glia. So you have an activated microglia, but also reactive astrocytes. And we studied this in a mouse model, and this is a very well-known Alzheimer's disease mouse model. It doesn't mimic the full disease, but it mimics the amyloid deposits. And here you see very nicely the amyloid in blue. You see that the microglia in red are moving into the amyloid deposits and are trying to degrade the amyloid. And you also see that the astrocytes are becoming much fatter when they are closer to the amyloid plaques. And we wanted to know what was changed in these glial cells, and we FACS sorted them out of the mouse brain tissue, and we did at that time a microarray. Nowadays you would do an RNA-seq, and we found several changes, which is here on this slice. So here you see the fold change in different genes in astrocytes in the first row, the fold change in different genes in microglia, and the more red the signal is, the more the bigger the changes. And what we mainly found is that the microglia showed an activation of the immune response, and that was expected because microglia are immune cells. But also the astrocytes showed an increase in the immune response. And the effect of the genes involved in immune-related genes was actually much more pronounced. So the increase was much more pronounced in astrocytes. We also checked on all kinds of different genes that are involved in this tripartite synapse, so that are involved, for instance, the glutamate signaling and glutamate recycling. And what we found was that the genes were almost all downregulated in Alzheimer astrocytes. And this is just to show you that we also found in astrocytes, for instance, that the immunoproteasome was upregulated. So these astrocytes somehow are not that much involved anymore in neuronal communication. So they get distracted, are more involved in the immune response. And that's what I think might lead to problems with cognition. So then you have this nice gene list, but you want to go towards function and also the functional changes in astrocytes. So we did calcium imaging, also in this case with the GCAM3, we're now working with GCAM6. We're also looking at care channels, we're looking at connectivity. And what we found is that in these Alzheimer mice, so these are hippocampal slices, and we looked at calcium transient in astrocytes. So here you see, so every trace is one astrocyte, and here you see increases in calcium. So that's the way how astrocytes communicate with each other. And what we found was that so the top two are wild type mice, and the bottom two are two Alzheimer mice of different age, 6 months and 9 months of age. What we did find was that we have more and longer calcium transients. So there is also a functional change in these astrocytes. So making a very long story short is that what we find in Alzheimer mice brains is that the glia, so that there's an activation of the immune response in the microglia, but also in the astrocytes. And there seems to be a reduced capacity of neuronal support, and we do see more and longer calcium transients in astrocytes. And we also have the feeling that astrocytes are more connected, although we are still working on that part. But what is needed is to make a translation to the human brain, because if we want to understand the biological mechanism, but also the pathology of brain diseases, but also of the developmental diseases, for instance, we need a good model of the human brain, and also in that model we could identify specific cellular phenotypes. So a lot of the work in the Alzheimer field, but also in other neurological and psychiatric diseases, is done on post-mortem human brain tissue that you can get from brain banks all over the world, also in different mouse models. But I think what we really need is a living system, so that, which is a bit more complex, so that we could also study the neuron-glia interactions, for instance. And the reason that we need that is that mice are not humans, and that was also mentioned by Idan Segev in the first talk. And during development, it's very clear that there are differences, because if you look in the rodent's brain during development, here is the zone where you have the stem cells and you have the radial glia, and the different layers of the cortex are being formed. And in the human brain, this is much more complex, and you do not only have a ventricular zone, but you also have a so-called outer subventricular zone. And this is really also needed to create this very complex cortical structures that humans have. On the other hand, there are also many indications now that, for instance, the glial cells in mouse and human are different, that the human astrocytes are much more complex, that they have very unique human features, and the same also goes for the microglia. So if you isolate microglia, or if you study microglia and you look at them with molecular biology, you can find core transcriptional signatures of human microglia. So it would really be nice to have this human system to work on. So one of the things that you can do if you want to translate that type of work to the human brain is that you also do, for instance, RNA-seq or other kind of analysis on human brain banks. And that's also what we started several years ago. So we isolated from human post-mortem brain tissues microglia and astrocytes, and we could do quantitative PCR on that. And you see here, for instance, that the astrocytes indeed expressed astrocytic genes such as GFAP and FGFR, and the microglia expressed microglia-specific genes such as TREM2 and CX3CR1. We did also RNA-seq on this, but a major problem if you isolate the cells like this is that the RNA quality is not very good. So we moved over the last years to nuclear RNA-seq, but then the amount of RNA in the nucleus is not fully representing of the RNA which is in the cells. So it is very interesting to look at these post-mortem brain tissues, but you can never manipulate the system. So therefore, we want to move to a more human model next to our mouse and post-mortem tissue. And what we want in a disease model is that the cells are alive, that the cells are from humans, and that there are multiple cell types in that there, and that you also have a 3D more complex system. And one of those systems might be human mini brains or cerebral organoids. So you see them here in this flask, and they are as large as a green pea. So they're quite complex, large structures, and they are very heterogeneous as well. So that makes it also a bit complex to work with them, but they are very interesting structures. Sorry. Now I'm going back. There it is. So these cerebral organoids were first published in 2013. A work from Madeline Lancaster and Jürgen Knoblich. And this makes it possible to start with human stem cells, for instance, from skin, so the induced pluripotent stem cells, and to make human disease models. And this is from their original paper. And you see that if you start with pluripotent stem cells, you can make embryoid bodies and you can push them in the neuroectodermal lineage, forming in the end cerebral tissue. And here they show an example of a retina that's being formed on one of the organoids. And here they have do some immunocytochemistry on one of these organoids. And you also see like hippocampal structures being formed. And here you see cortical layerings. So these mini brains are very interesting, and many people are very excited about them, but it's also not, of course, the answer on how to study brain diseases, because this is a developmental model, and meaning that if you want to study diseases like Alzheimer's disease, it is interesting, but it's not like an age model. So there are several pros and cons, as with any model, to also use this type of model. An important thing is that you can use actually stem cells from patients, and that is based on this iPSC development that was developed by Yamanaka. He got the Nobel Prize for that together with John Gurdon, and they can make from fibroblasts, but we also can do it now from blood cells or from hair cells. If you bring in a few transcription factors in these cells can fully go back to the embryonal stem cell state, meaning that you could make mini brains or cell cultures from all different patients and then also study them. And this is very powerful tool to work with. So how do you make organoids? So you start with those stem cells from a patient or from a control person. You can get them from skin or from blood. You reprogram them with these different factors, and then you get induced pluripotent stem cells. And you can grow them in vitro in different media, and they will form these embryoid bodies that you see here in these non-sticking well plates. And if you then just put them in a different type of medium and you get rid of the ROCK inhibition and get rid of the FGF, they will differentiate into a neuroectodermal structure. You put them in a droplet of matrigel and you can keep them alive for months, and then they form these mini brains. And in the original paper, they use like a rotating flask, and we use a so-called belly dancer because you have to constantly keep this cultures moving, because that somehow leads to the development of all these different structures in the cerebral organoids. And this is how they look. So we also do light sheet imaging, and here you see a structure like this, and all the red blobs are centers of cell clusters that are actively dividing. And here you see also different structures. You see that PAC6 is expressed, and CTIP2, which are all markers for the neuronal tissues, and CTIP2 is also a marker for specific cortical cell layers. So these are these structures. So they look like blobs. They have all kind of different things growing out of them, and we keep them in a on a belly dancer plate, so to constantly move them, and then we keep them there for weeks or even months. So these are not very quick experiments. It's quicker to work, I think, with transgenic mice than to work with these kind of structures. But of course, it's mimicking at least the human situation a bit better than the mice. So in these organoids, you see these centers where you have actually a mimicking of the subventricular zone. So there are the stem cells constantly proliferating, and they move out to form different layers, cortical structures in these cerebral organoids. And if you stain this for specific markers, we find neurons, astrocytes, also microglia, and there are also synapses in there, and these synapses are also electrical active. So we can measure minis in these organoid structures. I think it's very important to realize that this is a developing structure. So it's not a, as I said before, if you want to study Alzheimer's disease, it might be not the best model to use. So if you compare the molecular phenotype of these organoid structures, which you see here in days, 60 days, and this is the development of the human brain, then you see that the molecular phenotype of these organoids are early to mid-trimester human fetal brains. So are very early human fetal brains. They are not the adult human brains yet. So in these organoids, there are several cell types, and there are these centers of stem cells that are proliferating. And we've seen microglia and astrocytes in that, and I will show you in the next slides. The microglia were actually quite surprising, because what you do is that you push these embryoid body in the neuronal lineage, and microglia are from the hematopoietic lineage. So many people thought that microglia would not develop innately in these organoid structures, but we showed 2 years ago that they actually do. So if you look at astrocytes in these structures, and this is work not from our lab, but from Renner. So what they already showed is that if you follow these organoids over a long time, you see here, for instance, CTIP2 was already expressed quite early, but for instance, GFAP, one of the classical astrocytic markers, they come up quite late. And that's actually also what's happening during development. First you have the neuronal the neurons that are being formed. And then later on during development, you have the neuron to glial switch, and then you have the astrocytes being formed. So after 160 days in vitro, all organoids that we have checked so far have GFAP. And also here you see very nicely all these different layers in the cortical structures that are being formed in these organoids. So besides the cerebral organoids, there were several different papers where they showed 3D structures, where they looked at astrocytes, for instance, and a well-known one is from Pasca's group, but they made a kind of neural spheres, so they're not developing these different cerebral structures, but also in these spheres they found astrocytes, a lot of astrocytes. But they pushed it mainly in the astrocytic lineage. They do not have any microglia in there. And there's another report, Robert Krenchik et al. They showed that they could combine neurons and astrocytes, and then they form also these neural spheres or asteroids. And you see also nice development of the astrocytes in these spheres. But again, also these spheres do not have microglia. Microglia are very important if you think about studying all kind of different diseases. So there are 2 people in our department, or actually there are many more now because our organoid lab is really extended. And my colleague Jeroen Pasterkamp is working on ALS, and he actually started our organoid lab several years ago. And there are 2 people now working on astrocytes, and they simply cultured organoids, checked at several different time points in vitro with quantitative PCR in this case, to check when the glial markers would come up. And you see that you really have to wait quite a while, so 3-4 months before you see microglia marker increasing. And for instance, here GLT1 and GLAST1, which are the glutamate transporters, also important for neuronal functioning, they come up quite late. So this is day, I think, 180. So several different pictures here to show that also all these structures are developing over time, but there's also a lot of heterogeneity between the different organoids, but also between the different batches that we make. And here you see in blue all the different nuclei. You always see very nicely the areas where there's a lot of proliferation going on. And here you see also GFAP. In this case, we already found the protein at day 66. We see a very nice expression at the rim of the organoid, which is also the case if you look in normal brains. So there is a lot of GFAP at the outer areas of the brain. And if you zoom in, you see these radial glia-like structures. And at later time points, you see that these astrocytes also change shape and they become more like the astrocytes with the many processes that we know from that exist in the human and the mouse brains. So several more pictures, again, different days in vitro. We also checked S100 beta, which is an astrocytic marker, also here nicely at the rim. And in this case, also GFAP and S100 beta that co-localize in certain areas and certain cell types in these organoids. And at the higher magnification, but you also see there are cells only expressing GLT1 and there are cells only expressing GFAP and there are cells that are expressing both markers. So again, if you look at that in 3D with light sheet imaging, and this is, it says morphology of astrocytes, but these astrocytes are not filled with the fluorescent dye. So this is only based on the cytoskeletal, on the GFAP cytoskeleton, which is only about 10% of the whole volume of an astrocyte. But you see that there are several astrocytic areas in this organoid. And in this organoid, you see, again, the areas that are highly proliferative because they are Ki67 positive, so all kind of subventricular zones here, and vimentin present in these organoids. So the microglia story is a bit different and was also unexpected. because microglia, if you look through development, they are deriving from hematopoietic stem cells. So during development, during brain development, you have neural stem cells in these subventricular zones or the ventricular zone, and they form the macroglia, so the oligodendrocytes, astrocytes, and all kind of other glia, and they form the neurons. And the microglia actually enter the brain. They originate from the yolk sac. They are myeloid cells, and they enter the mouse brain at E8, so very early. And then once they are arriving in the brain, they start to differentiate into microglia. So they are from a different lineage, and therefore people always thought that you could not find them into these organoids. But these two people, Paul Hormel and Renata Baptista Vieira de Sar, they started studying that, and they simply stained our organoids with a microglia antibody, IBA1, and they found OMGs, they call them, so organoid-derived microglia in these organoid structures. And this is how it works during development. So what you see is that I think it's day 50. Let me see, yeah, day 52. So very early you see markers for early mesodermal cells within the developing organoid. So this is brachyury, and at this time point there's no IBA1 staining present yet. If you wait 10 more days, then at day 52 you see these microglia to appear, and there are still a few red stainings here. So they start in these clusters. So you always have these clusters of cells, and if you wait for a longer time, you see them spread all over the organoid. I also have to say that we do not always see it in every organoid, and that's depending on the donor, on the iPSC donor. But I think in 80% of the organoids that we've grown from several different donors, we do find these microglia develop. And it's not clear to us now why in certain donors we do not find the microglia development. And this is at a higher magnification. You also see that they're very nice. In the beginning, they're still quite roundish, or more monocyte-like. And if you wait longer, and also if this organoid is developing further, you see that all these microglia get these very nice processes, which are very similar to what you see in an adult human brain, for instance. So we also did RNA-seq on these microglia, and I do not have a picture in this presentation, but the molecular profiles of these microglia are much more similar to adult microglia isolated from human brain, and have a lower similarity to developing microglia. And here you see that there's also the perimeter, of course, becomes much larger if you look over time. These cells also express factors and genes that are known to be important for microglia development. So this interleukin 34, which is going up, CSF1, TGF-beta1, and also certain transcription factors like RUNX1, SP1, and CSF1R. And that's also what you see here. So this is all from our published work from 2 years ago, that the first bar are the microglia from the organoids, and the second bar are microglia that we isolate from adult human brains, so from post-mortem tissue. And you see that they are have quite similar expression levels, except for instance TREM2 and P2RI12 and TMEM119, and these are also classical microglia genes. But what we saw with these genes, that if you wait longer during development, you also see that these genes go up. So most likely the microglia need interaction with neurons and astrocytes in 3D to really start also expressing the genes that are also marker genes for these microglia in the adult brain. So we also checked whether these glial cells, these microglia cells, can also phagocytize. And we also checked what kind of proteins are expressed, because all the data so far that I showed here is all based on RNA, but we also did we did fluorescent, not sorting, but flow cytometry on surface markers, and then you see CD45, CD11C, all kind of markers that are expressed on the adult microglia are also expressed on these microglia that are derived from these organoids. You can also sort them out of the organoids and put them in culture. And this is the microglia from the organoids, and these are microglia from adult human brains. And on these microglia, we can also do functional analysis. And what we found, and this is still in the organoid, is that there's also an interaction with these microglia with neurons, because I showed you in the beginning of the talk is that microglia can prune synapses, and that is also happening during development. That's probably also why they are that early in the brain, so that if some of the synapses are, for instance, silent, then the microglia will prune them. And we checked for the presence of, so this is one microglia, and you see all kind of dots here, which is the postsynaptic marker PSD95. Here you also see close interactions with the microglia and neuronal structures in the organoid. And we did with STAT microscopy, we could prove that these PSD95 puncta are indeed within, so this is a higher magnification, they are in the microglia in our organoid. So this is indicating that these microglia in organoids are actively pruning. And as I said before, what we also checked is whether these microglia can phagocytize. So we put the adult microglia in vitro and we fed them with IC3B beads. And these beads are fluorescent, and you find these beads back in the microglia, but also the organoid-derived microglia can do that. So they are functionally active, they can interact with synapses, and they are most likely also pruning synapses in organoids. So in our organoid system, we have astrocytes and microglia, and we can also measure synaptic activity. So we make slices of these organoids, and we can measure traces from these structures. So these are electrical active. And what I just mentioned a few minutes ago is we also did RNA-seq on the microglia, and the microglia here are clustering together with adult microglia and are really different, of course, from fibroblasts and iPSCs, but also different from young developing microglia. So these brain organoids, I think, are very interesting structures for the future to study neuron-glia interactions, because we have alive cells. They mimic at least human brain development. So if you are interested in neurodevelopmental diseases, I think this is a great system. You can use patients' iPSC. So if you have working on a disease with a known genetic mutation, you can use that, and you can also use then a isogenic control by repairing the mutation with CRISPR-Cas9, so that you have exactly the same genetic background, and that's also, I think, a good model to use. These structures contain neurons and glia, but they also contain microglia, and that is, I think, very important for many of these diseases. And you can study 3D interactions. So I think these are very interesting tools in the future, but also now already, to study different brain diseases. But what I really would like to stress is that this is not the only solution. So if you want to study these interactions, I think you need different systems together. So you need the mouse models, you need human brains, but you also need these organoids, and it's depending on the question that you ask, which model will suit you to get your answer in the end. So I think it's a very nice tool, but you always need to use them with the mouse models, but also with the human post-mortem brain tissue. So this was my presentation. I would like to thank all the people in my lab and in the lab of Jeroen Pasterkamp that worked on the organoids. And this group is really extending now, because there are several people working on several different brain diseases now on organoids in our lab. And this is the group of people that with whom I work now for many, many years on reactive gliosis in Alzheimer's disease, but also in stroke. And These are my funding agencies. So this is the Dutch funding agency and an Alzheimer funding agency. So I would like to thank you for your attention. And if you have any questions, please go ahead.
So thank you very much for this fantastic talk. It's very interesting to see how much we can advance with these complementary systems, as you highlight in your last summary slide. So I would like to pass the word to participants, and there are actually 2 questions in the window, and I can see that there are also some hands-on. So let's start with Gertrude Perea, please.
Hi, Elly. How are you? Thank you for the great talk. I would like to look to the last part of the organoids process. And I was wondering, I mean, I guess this is kind of in the ongoing project, but I'm wondering if it's possible to use these organoids not only to study dysfunctions of or some diseases that you can tackle with a genetical approach, but also the senescence, the natural senescence of the brain. I mean, it's possible to use this approach like a faster life cycle of the brain to study what's going on from the development to the senescence of the brain?
Yeah, so we're trying to induce senescence by reducing the levels of lamin, for instance, B1, because it's also known that that's decreased in senescence. And you can also think about introducing these progeria genes. But so far, I mean, that's still ongoing work. And this is also work that I mean, it goes quite slow, right? Because you have to wait quite long before you have all the different cells, and then you have to age. But I think that you can use it to induce aging in vitro and then study how that would affect all kind of physiological functions. But I think you, as I said before, you should compare that also, for instance, with the work of Huib Mansfelder, who also does electrophysiology in human brain slices after operations. So all these different bits and pieces of information, I think, should come together in the end. But at least this system you can manipulate, and we can also work with all kind of fluorescent reporters, for instance. We can add GCAMs in there. We can do all kind of different things. But then we have to. So we are now busy in preparing a toolkit to look at all these neuron-glia interactions in the human iPSCs, so in the organoids. But that's something that we need so to move the field also forward.
Okay, thank you so much, Elly.
You're welcome.
So thank you, Gertrude. There are questions in the questions and answer chat, so I can tell you about this. Juan Garcia Leon. So I guess this is a methodological question. How do you perform immunocytochemistry of the whole organoids?
So there are several protocols for that. So you can so you work with all kind of soaps to get the antibodies in, and we make and we use Clarity, so iDisco and Clarity protocols, as you can also do on mouse brains and then we can do the imaging in 3D, but of course you can also freeze them, make cryosections, and then do imaging in 2D.
Okay. Second question is by Sergio Casas, and he thanks you for the presentation and have a question regarding whether tripartite synapses are present in the organoids.
I do not know, but I guess they will develop, but we have to think of a way how to study that. So maybe we should look at electron microscopy, but I would expect them to be there, yes.
Okay. So Beatriz Furones, she asked about whether you add microglia precursors to get microglial cells in the organoids. Yeah,
no,
can elaborate.
No, we don't. They don't do it. So they innately develop in the organoid system that we have. So we basically used exactly the same protocol as Lancaster and Knope did. Then we made one mistake with the concentration of heparin. And heparin pushes the neurons in the cells into the neuronal lineage. So we thought that that mistake was the cause of the fact that we have microglia in our organoids, but it's not, because we did a lot of tests, so it's not. So what we see is that in many of our organoids that we grow, we do have microglia, but sometimes we do not. And I know also other labs can repeat this, but there are also other labs that cannot. And I do not know exactly what it is. Maybe it's something that is typical Dutch in our lab or something like that. But we have it in, I think we have now over 20 or 30 different iPSC lines from controls, but also from different diseases where we do find these microglia. And we do not actively add them, so they develop innately in our microglia. Of course, you can also add them and then they go in, but the nice thing here is that they develop with the system. So if you're also interested in early pruning, for instance, you could potentially study that in this developing system.
Good. So 2 questions by Amaya Arangs. So how long do organoids can last?
Oh,
it's the first question. And the second, well, let's go with the first.
Yeah, I think up to 2 years we have tried now. Some of them, because what can happen is you get a necrotic core, because if they grow too big, then the nutrients from the cell culture medium cannot go into the core anymore. So we're now also playing in making microfluidic devices where we have channels inside the organoids so that we can make them, give them all the nutrients that they need, but we can keep them quite long. And the reason that we have to stop them, unfortunately, is very often because we get an infection, and that's very costly as well. But so we are trying to be very cautious and who is entering the lab and who is not, but we can keep them very long. Yes.
Good. So she also asked about what is the ratio of the neurons, astrocytes, microglia, and whether this ratio is conserved among the different IPCs lines.
We have not really studied this. We do see differences. So I think that we really should do stereotaxic analysis on different iPSC lines from different patients or from different individuals to see whether it's different. But as I said, in some cases, we do not find microglia. And that is still puzzling us why it's not working out like that.
Okay, so I think we can move,
last
move
question.
on.
There's one other question. Whether we can generate a
Okay.
BBB?
Yeah, I'm sorry, yeah.
No, the blood-brain barrier, it's I just submitted a project together with a blood-brain barrier person to connect the blood-brain barrier to organoids. If you have vasculature in the organoids, it's very often not a real barrier. So it's quite open. There are not so many tight junctions. And also the matrigel in which the organoids are embedded are attracting endothelial cells, and then you get like sprouting of these endothelial cells, and then you do not get a real blood-brain barrier.
Yeah, I can see that there is one hand on Beatriz Furones.
NG2 cells, yes, there are OPCs in there. We do at least see all the markers. We also see markers for parasites, but we have not looked into detail into these cell types yet. But we have not seen myelin formation yet, but I think that we really have to culture them for quite long to really find myelin formation, because this is a developing system and it would take quite long.
Hello, Elly.
Hi.
Hi. I'm the same one who asked about the precursors. I'm super amazed of your microglia appearance in the organoids, and looking forward for you to discover how is this happening. I just was wondering, because you looked at the phagocytosis capability. I was wondering if you tried, or you're planning to try to induce some kind of damage or induce damage in the organoids, and to see how the microglia react to it.
Of course. We tried LPS, And that's already in the paper. So we do find also effects of LPS. And we are now working with organoids, for instance, also for Alexander disease, which has GFAP mutations, but also for Alzheimer's disease. But there are several other groups working for that. And yes, a damage would be a good thing to try, or a stroke. Things. So we are thinking about all kind of direction. But as I said, it's not very often. Cell cultures are quite quick and dirty experiments. Cell lines, but in this case, it's not. It takes quite long and it's very costly. So you really have to think your experiments through very well. But of course, that's an obvious thing to try to see whether the microglia would be activated, and it would be really cool to do in vivo imaging on these organoids and see that the microglia would migrate towards a damage, right? That would be cool.
It would be amazing.
Yeah. But yeah, you're
Yeah.
welcome.
Okay, there are another couple of questions. Well, actually, another question is by Laura Lopez Mascaraque. So do you find NG2 cells in organoids?
Yeah, as I said, we find OPCs, so we find markers for that, but we have not looked into detail. But I think there are many different cell types in these organoids. But yeah, the markers are there, but we have not looked into detail.
Okay, so are there any further questions? We can keep discussing about these exciting tools. Actually, the relationship with functional characterization, whether the organoids are generating activities in the form of brain waves, for instance. I think this is a fantastic avenue to try to understand whether they acquire function. So we can move probably forward and continue this discussion in the at the end.
Yeah, thank you very much also for all the questions. Thank you very much.
Thank you.